Let's talk a little bit about animal reproduction and development, Chapter 30. There's two types of reproduction, asexual and sexual. Asexual reproduction doesn't require a partner. Basically, you just clone yourself. You get two identical or almost identical offspring, and there's lots of examples of animals that can do this. Black aphids, which are a pest on many plants, many of them are clones. Also, a lot of bacteria and single-celled organisms use asexual reproduction. It is a great way to get a lot of numbers up quickly. However, genetic variability is pretty limited. In sexual reproduction, you don't produce as many offspring as quickly, but because of gametes getting half your genetic code from one parent and half from the other, you increase genetic diversity, and that has a survival benefit in nature. Sexual reproduction, however, does require more energy, and so that's why you get fewer numbers. You have to attract females, breed them, and then raise the offspring. Now, it varies how much parental investment there is in offspring, but here is your basic. Now, this is human sexual reproduction. During meiosis, you take the number of chromosomes in one of your sex cells, a sperm in males, an egg in women, and it reduces them to half. They produce haploid gametes, meaning that humans have 46 chromosomes and 23 pairs. So sperm will only have 23 chromosomes, one of the chromosomes from each pair, as will eggs. So then when a sperm and an egg unite, you will have a diploid zygote, fertilized egg, which will ultimately, through mitosis, grow into an adult. So, sexually reproducing species, fertilization can be internal or external. Now, here you have sea urchins. There are several species of sea urchin, and they basically just release eggs and sperm into the water and let the current mix them and fertilize the eggs. This is external fertilization. Internal fertilization occurs when the males and females join together in some way and the gametes unite inside the body. Here are a pair of birds. The male will press his cloaca to the female's cloaca, and he will ejaculate sperm into the cloaca, which will then fertilize eggs. That's internal fertilization. Zygote begins to divide soon after fertilization is complete. The cells begin to differentiate or acquire specialized functions. Genes then determine the overall shape and structure of the animal's body in a process called pattern formation. We are learning more and more about this embryological development. There are two types of development. There's direct and indirect. In indirect development, basically you will have an immature or larval stage that will look different from the adult. In direct development, the animal is born as just a miniature adult and it just gets bigger. Good examples of indirect development would be caterpillars going into butterflies or tadpoles turning into frogs or toads. Human reproduction, if we look at that system a little closer, we've already talked about the diploid cells at fertilization, the zygote, and the haploid cells. And here's the male human reproductive system. Okay, you have the paired testes. This is where your sperm is manufactured. Also produces sex hormones. Sperm production occurs in these little coiled seminiferous tubules within the walls of the tubules. And so there's a germ layer inside of each seminiferous tubule, and it undergoes mitosis and then it will undergo meiosis and split into haploid cells. And these spermatocytes will divide meiotically, and you'll get eventually 4 meiosis-generated sperm. Takes about 74 days after the germ cell divided. Mature sperm cells are released into the lumen of the seminiferous tubule. The lumen is just the empty space. And here's kind of what happens. You have this germ cell layer. It produces a spermatocyte, which undergoes meiosis. And this secondary spermatophyte will become spermatids. The spermatids are haploid and then will become the sperm. So it's germ cell, primary spermatocyte, secondary spermatocyte, spermatid, sperm. So this kind of summarizes what I just said, showing what is going on. So in mitosis of the germ cell generates the primary spermatocyte, which will then produce, meiosis will then produce secondary spermatocytes, and meiosis 2 will produce spermatids. Each mature sperm has a haploid nucleus. There's a long flagellum that allows it to travel through the fallopian tubes to try to find an egg to fertilize. Now, mature sperm are stored in an area called the epididymis. And from the epididymis, they travel through what is called the Vas Deferens, a duct that goes just above the urinary bladder and connects with an ejaculatory duct. Now, the ejaculatory duct is shared in common. It's just the urethra with the urinary system. And so what happens, sperm come out of the epididymis, and they join up with the urethra. Now, the prostate gland, which kind of surrounds part of the urethra, first it will produce this alkaline fluid that will activate the sperm to swim. That's called pre-ejaculate. And the purpose of that is to neutralize the urethra. Urine is extremely acidic and will kill sperm. So this pre-ejaculate washes all that, any remnants or traces of urine out of the urethra so that it can not be a hostile area for the sperm. And then during the act of copulation, the male will ejaculate, and if he is inside the female, the sperm will travel down the urethra out into the vagina, get into the uterus, and then move towards the fallopian tubes. Hormones influence male reproductive function. Hypothalamus produces this gonadotropin releasing hormone. Anterior pituitary releases follicle stimulating hormone and luteinizing hormone. And these two hormones stimulate testes to produce testosterone. Now, females do the same thing. They produce eggs. However, their eggs are stored in the ovaries. And in the ovary, multiple eggs or one egg will come out. And that's called ovulation. And these little finger-like extensions on the opening to the ovarian tubes basically cause a current. So when the female ovulates an egg, it is sucked into this tube here, where it will meet up with sperm if it's going to be fertilized. Now, egg production, basically, every female is born with all the eggs she'll ever have or ever need. But they stop at an early spot, and then when it's time, once a month, she will ovulate during her reproductive periods, and one will become mature, and then it will be ejected from the ovary and sucked into this fallopian tube. The ovaries have over 1 million primary oocytes. These will become the egg. Each month after puberty begins, one oocyte completes meiosis 1. Each developing primary oocyte is in a fluid-filled follicle. Meiosis 1 yields a secondary oocyte that is released from the ovary. Ovulation. Meiosis also yields a pair of polar bodies. This is a haploid cell that contains almost no cytoplasm. Since eggs need to be larger, the cytokinesis stage of meiosis 1 and meiosis 2 is very unequal, meaning that by the end of meiosis in males, you would have one cell would turn into 4 haploid sperm. In females, one cell will turn into 1 egg and 3 smaller little cells called polar bodies that are unable to be fertilized, and they're just reabsorbed into the system. This allows the egg, if it's fertilized, to have a lot more resources. Secondary oocyte, arrested in meiosis 2, travels along the uterine tube, and if fertilized, it will become implanted in the side of the uterus. Okay, so let's look at the development of the egg in relation to meiosis. During mitosis, germ cells are duplicated and they will become primary oocytes. The oocytes during meiosis 1 will stop at the primary oocyte stage and wait. Then, when it gets into meiosis 2, it will become a secondary oocyte and it will end up producing polar bodies. The secondary oocyte is actually ovulated, and once it's ovulated, it can be fertilized. When it's fertilized by a sperm, you'll get a fertilized egg. Now, as part of this, basically, you get 3 polar bodies and 1 egg out of 1 oocyte. So, the egg's expelled from the ovary, it hits this fallopian tube, it travels down into the uterus. Now, the uterus has built a large lining with a lot of receptors in it, which will help it to undergo contractions when parturition or birth occurs. The lining is called the endometrium. Okay. Now, if the egg is not fertilized at the end of the cycle, the endometrium will be broken up and destroyed and then ejected during menstruation. If it is fertilized, the egg gets fertilized, it will implant into the uterus in the endometrium, and hormones will be released to maintain the lining so that the embryo can continue to develop. So the egg, it starts dividing as it travels along this uterine tube or fallopian tube, and here you can see it actually starting to embed in the endometrium. Now, this is dependent upon the proper hormones being produced. And these are the hormones. Primarily, the pituitary releases follicle-stimulating hormone and luteinizing hormone. This stimulates the ovaries to secrete progesterone and estrogen. The uterine lining is stimulated by these 2 hormones to maintain itself so it doesn't degrade and go into menstruation. And here's kind of your ovulation events. So you're developing follicle, you ovulate, it moves into the fallopian tubes. The follicle, where the oocyte was, will then start to degrade, and it will generate hormones to maintain the endometrium lining until the ovaries can produce enough hormones to keep the body from shedding this endometrium. Okay, it's called the corpus luteum. If the oocyte becomes, or egg becomes, fertilized. It's usually a 28-day cycle. Concentrations of all four hormones fluctuate over this cycle, and you can kind of see here ovulation tends to have a really high estrogen, helps stimulate that ovulation. And then whether or not you're pregnant will determine the fate of the endometrial lining. Now, follicle-stimulating hormones will start to increase in the bloodstream. Okay, here's your ovarian cycle. In the middle of the cycle, a spike in estrogen triggers a spike in luteinizing hormones, which causes the ovulation. And under the influence of the LH, the ruptured follicle will become the corpus luteum and will continue to release hormones to maintain the uterine lining if the egg is fertilized. The hormone is progesterone, and that makes this uterine lining get even thicker. And this prepares the uterus for possible pregnancy. If no pregnancy occurs, then the corpus luteum degrades after about 14 days. At that point, progesterone levels will drop because this will stop producing them, basically this corpus luteum. And what then happens is if you're not pregnant, you'll shed this lining. And if you'll notice, you'll see these blood vessels in there kind of coiled. Hormones will cause them to straighten out and coil up, and straighten out and coil up. That'll break up the lining. And then at a certain point, all of the blood vessels will relax. And by that time, all of the endometrial lining will be shed, and the blood will wash it away, and the whole cycle starts over again. Now, birth control. Lots of ways for birth control. Pills are a form that basically convinces your body that you're already pregnant so that you don't ovulate. But you have to take your pill correctly on every day. If you miss a pill, it will screw up the cycle, and then you run a risk of getting pregnant. There are also physical ways of eliminating the possibility of pregnancy. Other forms of birth control would be like a condom, even to the point of what is called interruptus, or pulling out before the male ejaculates. There is the rhythm method. In fact, here's a whole list of ways for birth control. If at some point you get to the point where you no longer wish to have children, males can get a vasectomy, females can get a tubal ligation. What that is, let me take you back up here and show you. Let's see, I need the Vas Deferens. Okay. Leading from the epididymis and the testes to the ejaculatory canal, which leads to the urethra, is this little tube called the Vas Deferens. You can have a chunk of that severed. Okay, and when it gets severed, it can no longer transfer sperm to the ejaculatory canal. Now, you will still have ejaculate because semen is a whole different thing. It basically is a fluid that carries the sperm where it needs to go. It also provides nutrients in the forms of sugars and things like that, so that the sperm can get where it needs to go. Now, a tubal ligation, let me find a blown up version perhaps. Here is the tube, the uterine tube. A tubal ligation just ties this off so that the ovulating eggs never reach an area where it can be fertilized. Let's see, there's withdrawal. There are implants which work like the pill, but you don't have to take a pill every day. And then there is the morning after pill, which will prevent an egg from being implanted in the uterus. Microbes that live. There are many different sexually transmitted diseases. Now, there's a lot of ways to prevent them. At the top of the list, if you never have sex, you'll probably never get a sexually transmitted disease. You can limit the number of sexual partners. Condoms are really good at preventing diseases being spread. You can get vaccines for certain things like Hep B and genital warts. These are some of the most common. On college campuses, chlamydia is probably the most common. And chlamydia, a lot of times, it's a bacterial infection, but it will be confused frequently with a urinary tract infection. Okay, so now, let's see. Syphilis, gonorrhea, chlamydia, trichomoniasis, and candidiasis. These can be cured. Genital herpes, if you get it, you will have it forever. At that point, all you can do is take medication to keep yourself from having outbreaks. Genital warts, very hard to treat, very hard to detect unless you actually have an outbreak. You can remove the warts. Best way to deal with that is just get the HPV vaccine when you're 12, which is when it's most effective. And then there's HIV or AIDS. Now, HIV is just an immunodeficiency virus that keeps your immune cells from functioning properly. And there are some treatments for it. It's very expensive to treat. The difference between HIV and AIDS is the number of functional T cells you have. As your T cells drop in your blood supply, once you get below a certain number, you then are considered to have AIDS. Fertilization to birth. So here is a sperm cell meets an egg. And there's a little structure on the end of the sperm called the acrosome that produces enzymes that allow it to penetrate. Now, once one sperm cell gets inside the egg, it depolarizes the whole surface, which repels all the other sperm cells. This keeps an egg from being fertilized by more than one sperm cell. The actual sperm is usually it is fertilized somewhere up here in the tubes, and then it comes into the uterus and it will latch onto the side of the endometrium and tissue will literally grow over it. And originally it will be in a cavity that will fill with blood, but eventually you will see blood vessels grow into the developing embryo. The embryo produces these structures that support its development, and you'll eventually get the umbilical, which will attach the uterus to the developing fetus. Okay, the amniotic cavity is just a cavity full of fluid, nutrients, waste products, etc. It's commonly referred to as the woman's water. So when you hear my water broke, this cavity full of amniotic fluid is pierced and all the amniotic fluid runs out. Okay. Now, the developing fetus here is basically receives its nutrients, oxygen, stuff like that through its umbilical. Sex difference becomes apparent between the 7th and 9th week. And so here would be an undifferentiated stage. Week 9, you can kind of see these are going to eventually be the scrotum and testes. And this will grow into a penis. Now, the sex-determining gene on the Y chromosome is activated and development continues as a male. Otherwise, female genitals will develop. At week 9, we call it the fetal stage. And fetus is not a derogatory term, or it does not imply no worth. It is just a timestamp. We divide everything in biology into phases. And so you're an egg, then you're a zygote, then you're an embryo, then you're a fetus, and then when you actually go through birth or parturition, you become a baby. This is so we can refer to the organism at various stages of development. There are diseases you can get in any stage. There are things that can go wrong in any stage. And so if you just use one term to apply to all of the stages, it makes treating and keeping this organism alive a lot more difficult. Okay? So we monitor it through all these different stages to produce healthy offspring. Birth, basically what happens during the entire time a woman is pregnant, she's building oxytocin receptors in the uterine lining. When it comes time to have birth, oxytocin is released, and oxytocin causes the uterus to contract and try to expel whatever is in there. As it does that, it causes even more oxytocin to be generated and released. It's a positive feedback mechanism. So each contraction increases the amount of oxytocin, which binds to more oxytocin receptors, which makes the contractions come quicker and stronger. Eventually, the head will crown. That means just the top of it will come out. It's going to press and open against the cervix. It goes through the birth canal. And then as it comes out, there's the umbilical. It will be attached to what is called the afterbirth. It just peels out the uterine lining with it. And once you get there, you have a baby once it comes out. Here is kind of that positive feedback system.